Aspirants, so here we are in our second session of biomolecules. Today we are going to deal with the complete biomacromolecules, carbohydrates, proteins, lipids and nucleic acids. Let's get started with carbohydrates. What are carbohydrates composed of? Carbohydrates are composed of carbon, hydrogen, oxygen. Their general formula will be CnH2nOn, right ma'am? Okay, it is CnH2n and ON. Okay, this will be the general formula. Fine, right? So here this is the composition of carbohydrates, and what are this commonly known as? These are commonly known as saccharides because they are the basic, their basic components are what? Sugars. Carbohydrates are composed of carbon, hydrogen and oxygen. They are commonly noted as what? Saccharides because their basic components are sugars. We have two types of carbohydrates, like small carbohydrates, large and complex carbohydrates. Small carbohydrates are again classified to biomicromolecules. Okay, biomicromolecules. They include monosaccharides, derived monosaccharides and oligosaccharides. These three comes under what? These three comes under biomicromolecules, whereas polysaccharides will come under biomacromolecules. Clear till here? Any doubt anywhere beta? And then we will move on to monosaccharides. These are the simplest carbohydrates. Monosaccharides cannot be further hydrolyzed. They cannot be further hydrolyzed into smaller components. We cannot hydrolyze monosaccharides into smaller components. They are composed just of 3 to 7 carbon atoms. They are just composed of 3 to 7 carbon atoms. They are known as biomicromolecules. For example, if you see glucose, fructose, mannose, galactose, these are what? Hexoses. And then you see glucose. Glucose is known as dextrose or grape sugar or corn sugar also. Fine, right? Glucose is also known as dextrose, grape sugar or corn sugar. Glucose is the universal sugar, remember. Okay, glucose is the universal sugar. Fructose is known as fruit sugar or livulose. Okay, fructose is known as fruit sugar or livulose. It is the sweetest among the naturally occurring sugars. Remember that. Fine, right? Which type of monosaccharides is sweetest among all them? Fructose. Remember that. Fructose or livulose, this is the sweetest among naturally occurring sugars. Monosaccharides, they will have free aldehyde and ketone group to form glycosidic bonds and to enhance disaccharides. Okay. So, to turn up into disaccharides, they will be having free aldehyde and ketone groups. That is why they are addressed as what? Reducing sugars. If you can see the structure of glucose and fructose over here, here you can see free aldehyde and ketone groups. Here can you see free aldehyde and ketone groups are present. If you can observe for fructose, glucose, they will be connected to other monosaccharide by N-glycosidic bond. Fine, right? So, as they are having free aldehyde and ketone group, they are addressed as what? Reducing sugars. This is all about monosaccharides. Then we will move on to oligosaccharides. Oligosaccharides, these are small carbohydrates. Oligosaccharides are small carbohydrates. They are formed by condensation of 2 to 9 monosaccharides. Remember that. They are formed by condensation of 2 to 9 monosaccharides. Monosaccharide units are further joined by glycosidic bonds. As I told you, monosaccharides are joined by glycosidic bonds because of their free aldehyde and ketone groups. Next, glycosidic bond, here it is usually this forms between what carbon's glycosidic bond is formed? Between carbon, first carbon and fourth carbon of the neighboring sugar units. Fine, right? Between first carbon and fourth carbon of neighboring sugar units, glycosidic bond will be formed. Followed by, this is based on number of monosaccharide units. If you see disaccharides for example, sucrose, maltose, lactose and trehalose. Trisaccharides include raffinose, which is made up of glucose, fructose and galactose. Next, tetrasaccharides include stachyose. Okay. Tetrasaccharides include what? Stachyose. Remember that. These are what? Oligosaccharides. Followed by, so here you can see the disaccharide units. Disaccharide units, just a sec. Yeah, disaccharide units. If you see maltose, maltose is made up of 2 glucose units, right ma? 2 glucose units, and this is by 1,4 glycosidic linkage between 2 monosaccharides. Fine, right? Maltose is a disaccharide. Then you see lactose. Lactose is a disaccharide. What are the monosaccharides that are present over here? Glucose and galactose. Again, this is 1,4 glycosidic linkage. Next, sucrose is a disaccharide which is having 2 monosaccharides, again glucose and fructose. Okay, that is alpha 1,4 glycosidic bond, alpha 1,4 glycosidic linkage. Clear, right? Maltose, lactose and sucrose. Clear till here? Next you see polysaccharides. Polysaccharides are long chains of sugars. They are threads that contain different monosaccharides as building blocks. So, here these are obtained where in acid insoluble pellet. They have polysaccharides. Again, how polysaccharides are built up? By N-glycosidic bond. N-glycosidic bond is formed by dehydration. N-glycosidic bond which is formed by dehydration, that is removal of water. Fine, right? And how the linkage occurs in polysaccharides. In the structure of polysaccharides, monosaccharides are the building blocks over here. These individual monosaccharides are linked by what? Glycosidic bonds. These individual monosaccharides are formed by glycosidic bonds. The glycosidic bond is formed by dehydration. Remember, it is formed by what? Dehydration between 2 carbon atoms. This 2 carbon atoms of 2 adjacent monosaccharides. Clear? These are the linkages that you will see in polysaccharides. Clear, right ma? This is about monosaccharide, disaccharide, oligosaccharide and polysaccharides. Now we will see types of polysaccharides. Important, many at times the questions are asked from cellulose and starch. Okay, that is homopolysaccharide and heteropolysaccharide. We will learn that in detail. Look into this ma. Depending on the composition of polysaccharides, polysaccharides are of 2 types. We have 2 types of polysaccharides. Depending on the composition, polysaccharides are of 2 types. One is homopolysaccharide, second will be heteropolysaccharide. Homopolysaccharides and heteropolysaccharides. Homopolysaccharides, these are formed by polymerization. They are formed by polymerization of one type of monosaccharide monomer, and heteropolysaccharide, this is formed by condensation of 2 or more types of monosaccharides and their derivatives. Remember the examples properly. Examples of homopolysaccharide will be starch, chitin, glycogen and cellulose. Fine, right? Heteropolysaccharides include peptidoglycan and agar. Fine, right? Next, if you see cellulose over here, this is a polysaccharide. This consists of only one type of monosaccharide. Cellulose is a homopolymer of what residues? Glucose residues. They are joined by beta 1,4 glycosidic bonds. Fine, right? They are joined by beta 1,4 glycosidic bonds. The plant cell wall is composed of cellulose. Plant cell wall is composed of cellulose. Paper is made from plant pulp and cotton fiber is again cellulosic. Paper is made from plant pulp, cotton fiber is cellulosic. Cellulose does not contain complex helices. They do not contain complex helices. That is the reason they cannot hold iodine. They do not respond to iodine test. Fine, right? So, what are the keynotes that you have to remember from cellulose ma? Cellulose is a polysaccharide. It consists of only one monosaccharide units. It does not contain complex helices, as such it will not hold iodine. Fine, right? And these are the direct liners of your NCERT, where they are occurring. Cellulose is the main composition in the plant cell wall. Of course, plant cell wall will also consist of cellulose, hemicellulose and pectin, but cellulose is the main composition in the plant cell wall. Paper is made up of plant pulp and cotton fiber is again cellulosic. Till here everything is clear right now, and then we will see starch. Starch is a homopolymer of alpha D glucose. Fine, right? Starch is a homopolymer of alpha D glucose. Starch is composed of two types of polysaccharides. One is amylose and the second is amylopectin. It consists of what beta? Amylose and amylopectin. Amylose is a straight chain polymer. It is a straight chain polymer of alpha D glucose units. It is a straight chain polymer of alpha D glucose units where they are linked up by alpha 1,4 glycosidic bonds. Fine, right? Amylopectin is, this is again branched chain polymer of alpha D glucose units which consists of two types of bonds. One is alpha 1,4 glycosidic linkage and second will be alpha 1,6 glycosidic linkages. And then 80% of starch is consisting of amylopectin itself. And here starch possesses complex helices because of alpha 1,4 and alpha 1,6 glycosidic linkages of amylose and amylopectin. This consists of complex helices and this will give what? Blue color for starch iodine. Starch and iodine composition, this will give out what? Blue color. This will be blue in color. So, this is about starch. Fine, right? Go through this. If you are having any doubt, just let me know. Here we go with glycogen. Glycogen is another polymer of alpha 1,4 glycosidic bonds and alpha 1,6 glycosidic bonds. The right end is called as reducing end and the left end is addressed as non-reducing end. Fine, right? Right end is known as reducing end and left end is known as non-reducing end. This is for glycogen. And then here you can see this is the direct diagram from your NCERT, wherein here you can see right end is the reducing end and left end is non-reducing end. This is non-reducing end. It does not have free aldehyde and ketone groups, whereas here you can see the free aldehyde and ketone groups. Fine, right? Next. Chitin. Chitin is found as structural component of fungal walls. Okay, chitin is composed of chitin is found as structural component of fungal walls, and this is also seen in exoskeleton of arthropods. Okay, exoskeleton of arthropods. Remember, this is direct NCERT liner. Where do you see chitin composition? In fungal walls and in cytoskeleton of arthropods. The basic unit of chitin, chitin has what units ma? NAG units, N-acetyl glucosamine units. Okay, they consist of N-acetyl glucosamine units and this is an unbranched polysaccharide. This is what? Unbranched polysaccharide. These are joined together by alpha 1,4 glycosidic bonds. Fine, right? These are joined together by alpha 1,4 glycosidic bonds. The monomers of N-acetyl glucosamine are these glucosamine units, like glucose units. Fine, right? So, chitin, where is it present and what are the units that are present? N-acetyl glucosamine units, and they further consist of monomers, those are glucose. Next, inulin. Inulin, this is a polymer of what? 1 monosaccharide, that is fructose. Inulin is a polymer of fructose. The source of inulin is found in roots and tubers of certain plants like dahlia, chicory, Jerusalem artichoke, asparagus and onion and garlic. These are the sources of inulin. Next, biological role. Inulin acts as storage polysaccharide. Remember, inulin acts as storage carbohydrate in some plants. It acts as storage carbohydrates in some plants, and it is not digested by human. Okay, this is not digested by human digestive enzymes. And what are the uses in human physiology? The uses include, like this is used to measure glomerular filtration rate in kidney functioning, fine, right, and it is freely filtered by glomerulus. It is not absorbed, secreted or metabolized by kidney. Hence, inulin clearance will be equal to glomerular filtration rate. Inulin clearance will be equal to what? Glomerular filtration rate. Very important, this is not there in your NCERT, but for your knowledge I am telling you. Inulin is, this is not absorbed by the human system, so this will be removed off. That inulin clearance will be equal to glomerular filtration rate, where you will learn clearly in excretion of your human physiology. Till here it is clear right now, and then we will move on to proteins. Yeah. So, carbohydrates, what we have learned, its characteristic features: monosaccharides, disaccharides, oligosaccharides, polysaccharides, and then we have learned homopolysaccharides, heteropolysaccharides. We have also looked into cellulose, starch, chitin, and at last inulin. Fine, right? Now, we will move on to proteins. Proteins are polymers of what? Amino acids. Proteins are polymers of amino acids. A protein is a heteropolymer, it is not a homopolymer, remember that. A protein is a heteropolymer, it is not a homopolymer, because you will have different types of amino acids. As such, 20 different types of amino acids. These different types of amino acids will make up a polypeptide chain. Fine, right? This will make up a polypeptide chain. As a polypeptide chain is made up of different amino acids, we say these are heteropolymers but not homopolymers. If you see the structure of a single amino acid consists of an N-terminal and a C-terminal, right now? N-terminal and a C-terminal, that is, it has an amino group and a carboxyl group, hydrogen binded to this and an R group. All amino acids will be variable based on different R groups, whereas all the amino acid will consist of amino, carboxyl group and hydrogen group. Fine, right? They will be variable in terms of what? R group, that is functional group. Here we go with the basic classification of amino acids, where you have to fix this in your mind, very important. Amino acids are primarily classified based on their polarity. So, if you see, based on their polarity, amino acids are classified into what? Hydrophilic polar amino acids and hydrophobic non-polar amino acids. Hydrophilic polar amino acids, we have both the types of charges, negatively charged and positively charged amino acids. Negatively charged amino acids, negatively charged polar amino acids include aspartic acid and glutamic acid. Positively charged polar amino acids include what, ma? Lysine, arginine, histidine, which are most noted we know, followed by serine, threonine, asparagine, glutamine and cysteine. Fine, right? And then hydrophobic or non-polar amino acids. Hydrophobic or non-polar. Here we have two sets, aliphatic side chain and side chain. Aliphatic includes glycine, alanine, valine, leucine, isoleucine, proline and methionine. Followed by aromatic. Aromatic hydrophobic amino acids include phenylalanine, tryptophan and tyrosine. If you fix this chart in your mind and look into it twice and thrice, okay, that will be easy to you, okay, and that is very important also. Fine, right? Just go through this. Take a screenshot if you want. Fine, right, ma'am. And next we will go with, yeah, essential and non-essential amino acids, and these are the amino acids which are given in your text. So, here we see the basic amino acids like, you know, glycine, alanine and serine. These are the amino acids where glycine is not optically active because it does not have a functional group, it just has H group. In the R group section, it just has H. Next, alanine has methyl group, serine ethyl alcohol. Okay, so it has CH2OH group. Fine, right? Methanol, sorry, methanol, and here it is methyl group for alanine. Here, if you see essential and non-essential amino acids, to remember essential amino acids, I will give you a mnemonic. PVT, Tim Hall. PVT, Tim Hall. Okay, private Tim Hall. This also you can remember as such. So, this is what private Tim Hall or PVT Tim Hall, where P stands for phenylalanine. Okay, P phenylalanine, V valine, tryptophan, threonine and I isoleucine, then M methionine followed by histine, arginine, leucine, lysine. Clear? Phenylalanine, valine, tryptophan, threonine, isoleucine, methionine, histidine, arginine, leucine and lysine. Till here it is clear, right, ma? Essential and non-essential amino acids. Now, we will move on to zwitterionic forms. What is a zwitterion? Zwitterion is the major segment that you have to focus when you are going through proteins, because n number of times the questions have been asked from zwitterion. Firstly, what is a zwitterion? Zwitterion is a molecule that is present in the neutral pH, where the amino acid will have both cation and anion. Okay? So, it is a molecule with functional groups of which at least one has positive charge and one has negative charge at neutral pH. If you reduce the pH from the zwitterion, if the pH is reduced from zwitterion, it consists of only cation. If the pH is increased, it has only anion. At neutral pH only the amino acid exhibits both anionic form and cationic form. Here you see this table clearly with a, at low pH, that is at acidic pH, it has only cation. The structure is amino group and carboxyl group, where the net charge is +1. Neutral pH, at neutral pH, zwitterionic form exists where it has both amino group and carboxyl group, where the net charge is 0, followed by high pH, that is basic pH. It has only anion. Cation is not present over here, where the net charge will be -1. Till here everything is clear, right now, zwitterionic form, and then you go through, yeah, peptide bond over here. The peptide bond between amino acids is formed. The peptide bond between amino acids is formed when carboxyl group of one amino acid, it will react with amino group of the other amino acid. Can you see here clearly, ma? Yeah, carboxyl group of one amino acid will react with amino group of the next amino acid, forming what? A peptide bond. The first amino acid will have N terminal and the last amino acid will have C terminal. As such, we have many structures or levels of proteins. Fine, right? So, first you have to know how a peptide bond is formed between two amino acids. Okay, peptide bond, how is it formed between two amino acids? It is a bond between carboxyl group and amino group, and first amino acid will have free N terminal, last amino acid will have free C terminal. Remember these statements carefully, because we can expect statement based questions. When statement based or assertion reason questions are asked, you will get confused again. So, remember the statements clearly. These are direct NCERT liners. The first amino acid is the N terminal amino acid, last will be C. Protein structures at 4 levels you see. We have 4 levels of protein structures: primary, secondary, tertiary and quaternary. 4 levels of protein structures we have. Fine, right? First we will see the primary structure of protein. This is the primary structure and these two include the secondary structures. Secondary structures will have alpha helix and beta pleated sheets. So, here you see the sequence of amino acids in the primary structure. In the primary structure, the sequence of amino acid will give positional information in the protein. Okay, this will give what? Positional information in a protein, which is the first amino acid and which is the last amino acid. That positional information is given by the primary structure. The first amino acid which is second. Fine, right? So, from the primary structure what we will get to know? Positional information. A protein is imagined as a line. The left end is represented by the first amino acid and the right end is represented by last amino acid. Clear, right? The first amino acid is also called as N terminal amino acid and the last amino acid is also addressed as C terminal amino acid. I hope the primary structure is clear to you, right? Primary structure is a long chain. Fine, right? What do you know from the primary structure? Positional information. That's it. Next. So, here we will move on to the secondary structure of proteins. Secondary structure of proteins. In proteins, only right-handed helices are observed. Remember, what are observed, beta? Only right-handed helices are observed, like alpha helix and beta pleated sheets. Alpha helix and beta pleated sheets. In an alpha helix, the carbonyl of one amino acid is hydrogen bonded to H of the amino acid of 4 down carbon. Can you see here? Carbonyl group of one amino acid is binded to amino group to 4 down amino acid. As such, it is forming what? As such, it is forming a helical structure like this. Fine, right? Because carbonyl group and amino group are connected 4 down chain. Next, this pattern of bonding pulls the polypeptide chain into a helical structure or a helical strand like this. Here what bond is present, ma? Hydrogen bond. Okay, and then beta pleated sheets. In a beta pleated sheet, 2 or more segments of polypeptide chain, they will line up next to each other. Here can you see 2 or more polypeptide chains, they will line up next to each other, forming up what? Sheet like structure. They are held together by what bonds? Hydrogen bonds. So, here one point you have to highlight, that in the secondary structure of proteins, what are the bonds that are present? Beta, hydrogen bonds. And next you see, yeah. In addition, the long protein chain is also folded. This long protein chain, either alpha helix or beta pleated sheet, it is further folded into a woolen ball like structure, giving you what? Tertiary structure of protein. Tertiary structure of protein. Here you see tertiary structure. This is 3-dimensional structure of protein. Fine, right? This is 3-dimensional structure of protein, and here tertiary structure is absolutely necessary for many biological activities of the proteins, wherein R group interactions over here include hydrogen bonding, ionic bonding, dipole-dipole interactions and hydrophobic interactions as well. If you see the secondary structure either in the alpha helix or beta pleated sheets, only hydrogen bond is present. Whereas here if you see, you have in tertiary structure along with hydrogen bonding, you have ionic bonds, dipole-dipole interactions and hydrophobic interactions as well. Clear till here, ma? Any doubts anywhere? Yeah. Now, quaternary structure. Quaternary structure. So here, quaternary structure, best example I can give you is hemoglobin. Best example I can give you is hemoglobin. Here, some proteins assemble of more than one polypeptide or subunits. Proteins assembly, they will have more than one polypeptide or subunits. Hemoglobin is one example. Okay, hemoglobin is one example of this quaternary structure. Adult hemoglobin will consist of 4 subunits. 4 subunits include what, ma? 2 alpha and 2 beta subunits. 2 alpha and 2 beta subunits. Clear, right? Yeah. Next. So I hope you are clear with different types of amino acids first, then bitter ionic form. Different types of amino acids include hydrophobic, hydrophilic, essential, non-essential amino acid, followed by bitter ion structure, and then 4 levels of protein structures like primary structure, secondary structure, tertiary structure and quaternary structure. Now we will move on to different types of proteins and their functions. Clear, ma? So here we go. Here we go with this proteins and their functions. Most of your body is composed of proteins in these forms like collagen, which is the second abundant protein after rubisco, and most abundant protein in the animal world is collagen. This will form intercellular ground substance. Next is trypsin. Trypsin is again an enzyme. Insulin. Insulin is working as what? Hormone. Antibodies fights against infectious agents. Receptors, these are sensory in reception like smell, taste and hormones. Next, GLUT4. This will enable glucose transport into the cells. GLUT4 will enable what? Glucose transport into the cells. As such, we have different types of proteins and their functions. Fine, right? So here you see functions of proteins. Some proteins will transport nutrients across the cell membrane. Some are hormones and enzymes. Some will fight against the actions like antibodies. And the most abundant protein in the animal world, that is collagen, is again a protein. Rubisco is the most abundant protein in the whole biosphere. Fine, right? These statements are very important, ma. And see what is the diverse roles of a proteins in a organism. Clear, right? It can act as an enzyme, hormone, antibody, receptor, ligand and collagen. Fine, right? Next. So after proteins, we are left with 2 biomacromolecules. Those are nucleic acids and lipids. Just go through this and then we will start about nucleic acids and lipids. So here if you see, ma, along with polysaccharides and polypeptides, nucleic acids also comprise of true macromolecular fraction. Okay, they will also comprise of what? True macromolecular fraction in any living tissue. Fine, right? And what are the building blocks of this nucleic acids? Building blocks of the nucleic acid is nucleotides. 3 chemically distinct components are present. One is the heterocyclic compound, next second is a monosaccharide, and third is a phosphate. Here I can show you the example and tell you. So, only sugar and nitrogen base will make it a nucleoside. Fine, right ma? Sugar and nitrogen base will make up a nucleoside. Nucleoside along with phosphate group will make up a nucleotide. So, you should know different types of sugars. Here deoxyribose is mentioned, but we have 2 types of pentose sugars based on the type of nucleic acid. It can be deoxyribose nucleic acid or ribose nucleic acid. In both types of nucleic acids, you will have ribose sugar. Ribose sugar is pentose carbons. Fine, right? So, here the nucleotide, for example, adenine nucleotide. Now, you got to know you have 2 types of pentose sugars, one is deoxyribose, second is ribose. What is the primary structural difference between deoxyribose and ribose sugar? Second carbon OH group. Second carbon OH group is present for ribose sugar. Second carbon OH group is absent for deoxyribose sugar. It is present in ribose and absent in deoxyribose. That is the primary structural difference between deoxyribose and ribose. What type of nitrogen bases you have? We have 2 types of nitrogen bases, purines and pyrimidines, right ma? Purines include adenine and guanine, pyrimidines include cytosine, thymine and uracil. Cytosine and thymine will be present in DNA and cytosine, uracil will be present in the RNA. Fine, right? And what are the bonds that are connecting between sugar and nitrogen base, sugar and phosphate group to form up one nucleotide? I will explain now. Okay, the heterocyclic compounds in a nucleic acid are nitrogenous bases like adenine, guanine, uracil, cytosine and thymine. Till here it is clear, right now? Next. Yeah, these are the diagrams that are given. If you see here, sugars. Yeah, as I told you, you have 2 types of pentose sugars. 2 types of pentose sugars. One is ribose, it has at second carbon it has OH group. Second is deoxyribose. Okay, deoxyribose, sorry, this is glucose, right? Glucose, it is hexose. Second is deoxyribose, it does not have OH group. Clear, right? These are the 2 pentose sugars. Now, we will speak about nitrogen bases. We have purines and pyrimidines. Let us talk, pyrimidines include what? Thymine, cytosine, uracil. And purines include adenine and guanine. Here it has 2 cycles, heterocyclic, it has total 9 nitrogens, it is heterocyclic. Whereas pyrimidines, it has just 1 cycle or 1 ring which is homocyclic, right. So, if pyrimidine nitrogen base is adjoining with the sugar, okay, adenosine which is ribose sugar, right. Ribose sugar is having OH group over here, adenine is binding. Purine is binded to sugar where N9 of purine is binded to C1 of the carbon by what bond? N-glycosidic bond. Remember, N-glycosidic bond. Fine, right. Now, here we have C1, C2, C3, C4 and C5. To 5th carbon of this nucleoside, phosphate group is attached. Phosphate group is attached to 5th carbon of the nucleoside by what bond? Phosphoester bond. Phosphoester bond. So, in a single nucleotide you will have two bonds, one is N-glycosidic bond and second is phosphoester bond. This will form one nucleotide. One example is adenylic acid. Adenine is the nitrogen base, it binds to the sugar forming nucleoside. Nucleoside along with the phosphate group at 5th carbon, this forms a nucleotide. Best example is adenylic acid. Pyrimidine to the nitrogen base. Let us see, uracil to the sugar. This forms out what nucleoside? Uridine. Okay, this is homocyclic. When pyrimidine is binding to the sugar, what type of bond is there, ma? C1 of sugar anyhow, this binds to N1 of the nitrogen base by again N-glycosidic bond. N-glycosidic bond. This you will get more clarity in mol bio again. Okay. So, clear, right? How a single nucleotide is formed? Single nucleotide is formed by sugar, nitrogen base, phosphate group. Sugar binding to purine, it will be C1 N9 glycosidic bond. Sugar binding to pyrimidine will be C1 N1 glycosidic bond, and sugar binding to the phosphate group at 5th carbon is phosphoester bond. So, in a single nucleotide you will have one glycosidic bond and one phosphoester bond. Clear, right? So, these are the purines and pyrimidines. Make a clear note of this beta, make a clear note of this. Yeah. Now, we will see the synonyms of this nitrogen bases as well. What is a common name and how this nitrogen bases are linked up, we will discuss till here, and silent features of DNA, different types of DNA and lipids we will look in the next class. So, here you see first adenine, it is also addressed as 6-aminopurine. Adenine is also addressed as what? 6-aminopurine, it is found in DNA and RNA. Guanine is also known as 2-amino-6-oxypurine. Okay, it is found in DNA and RNA. Cytosine also known as 4-amino-2-oxypyrimidine, found both in DNA and RNA. Thymine is also known as 5-methyl-2,4-dioxypyrimidine, also found in DNA, and uracil also known as 2,4-dioxypyrimidine, this is found in RNA. Take a screenshot of this, ma, very important. Okay. What are its systematic name or IUPAC name, you have to know about the nitrogen bases. Done? Clear till here? So, in our next class we will wind up the biomolecules, and thereafter we will solve the EAPCET PYQs from biomolecules, which ends the unit cell biology, and then we will move on to sexual reproduction of flowering plants from your text 1. Fine, right? So, till here everything is clear, no ma? So, with this we are done with today's session. In our next session we will move on to silent features of DNA, Chargaff's rule, double helical model of the DNA, and then we will also look into the lipids, different types of lipids as well, different types of DNA and enzymes in detail. We will wind up this chapter in our next class. Okay. Till then, look into this class, revise till here, and then we will start up tomorrow. Okay. So, that is all for today's session beta. Thank you.